Topic: Cells, the basic unit of life. Aims of lesson. At the end of this lesson, you should be able to know the following cell structures: mitochondria, ribosomes, plastids, vacuole, lysosomes, vesicles. Differences between plant and animal cells. Introduction. Revise organic and inorganic compounds that were covered in lessons 2, 3, and 4. Revise cell structure and function that were covered in the Week 5 lesson. Cells are mostly made of proteins, carbohydrates, lipids, nucleic acids, and water. Cells differ in size, shape, and structure in order to carry out specialized functions. Cells contain organelles with structures adapted to perform specific functions within the cell. Cells are very small, and it is through the development of microscopic techniques that we have been able to visualize and understand them. Concepts and skills. Below are definitions of some of the terms that you will study under this section. Use the glossary in your textbook to define these new terms/words. Note: at least 2 marks are awarded if you can define a term correctly in a test or examination. Chlorophyll, the green light-trapping pigment found in the leaves. Tonoplast, the membrane surrounding the vacuole in cells. Chloroplast, the organelle in the cytoplasm of plant cells that is responsible for photosynthesis. Turgor pressure, the firmness in a plant cell due to the pressure of water in it. Endoplasmic reticulum, the membrane system in a cell on which ribosomes sometimes occur. Leucoplasts, colorless plastids that store food in an insoluble form. Study the following information on cell structure and function. The roles of organelles. Mitochondria. A mitochondria occurs in plant and animal cells and are found in the cytoplasm. It is cylindrically shaped and is enclosed by a double membrane. The inner membrane contains folds known as cristae. The cristae increase the inner surface area of the mitochondrion where chemical reactions take place. The mitochondrion is filled with a semi-fluid substance, the matrix. Function. The mitochondrion releases energy during cellular respiration. Ribosomes. Ribosomes are small round structures that occur in plant and animal cells. Ribosomes may occur singly in the cytoplasm or in groups or may be attached to the endoplasmic reticulum, thus forming the rough endoplasmic reticulum. It consists of RNA and proteins. Function. Ribosomes are the sites of protein synthesis. Endoplasmic reticulum, ER. The ER is a membranous network found in plant and animal cells. There are 2 types of ER, i.e. rough ER has ribosomes on its surface, and smooth ER has no ribosomes on its surface. Function. The ER transports substances from one part of the cytoplasm to another. Golgi body. The Golgi body consists of a stack of flat membrane-bound sacs called cisternae. It is found near the nucleus and endoplasmic reticulum. It occurs in plant and animal cells. Functions. The Golgi body plays a role in producing and processing secretions, e.g. mucus and saliva. It prepares proteins for transport to other parts of the cell. Plastids. Plastids only occur in plant cells. There are 3 types of plastids, i.e. chloroplasts, chromoplasts, and leucoplasts. Chloroplasts. Chloroplasts are mainly found in the photosynthesizing parts of a plant, e.g. the leaves. A chloroplast is surrounded by a double membrane. A fluid matrix, the stroma, fills the chloroplast. In the stroma are disc-shaped membranes, the lamellae or thylakoids. The lamellae form small stacks, the grana. The chlorophyll, green pigment, is embedded in the lamellae. Chromoplasts. Chromoplasts occur in yellow, orange, and red flowers, leaves, and fruit. They contain pigments called carotenoids. Leucoplasts. Leucoplasts are colorless plastids that occur mainly in cells that store food, e.g. in potatoes. Functions. The process of photosynthesis occurs in the chloroplasts. Chromoplasts give the yellow, orange, and red color to flowers, leaves, and fruit. Leucoplasts store food. Vacuole. Vacuoles are fluid-filled organelles that occur in the cytoplasm of most plant cells, but are very small or completely absent in animal cells. A vacuole is enclosed by a selectively permeable membrane called the tonoplast. Vacuoles are filled with a liquid called cell sap consisting of water, mineral salts, sugars, and amino acids. Functions. The vacuole plays an important role in digestion and excretion of cellular waste and storage of water and organic and inorganic substances. The vacuole takes in and releases water by osmosis in response to changes in the cytoplasm, as well as in the environment around the cell. The vacuole is also responsible for maintaining the shape of plant cells. When the cell is full of water, the vacuole exerts pressure outwards, pushing the cell membrane against the cell wall. This pressure is called turgor pressure. Other types of vacuoles. Vesicles, small, spherical sacs which facilitate the metabolism, transport, and storage of molecules. Lysosomes, small vesicles, mainly found in animal cells, that are filled with digestive enzymes. These powerful enzymes can digest cell structures and food molecules such as carbohydrates and proteins. Differences between plant and animal cells. Know the meaning of instructional verbs in test and examination questions, e.g. Answer the following questions. Question 1. 1. The diagrams below representing two cells with their organelles. 1.1 Which one of these diagrams, X or Y, represents a plant cell? 1.2 Give two visible reasons for your answer in question 1.1. 1.3 Name the process in cell X where B plays a role. 1.4 Write down the letter in cell Y that can be associated with each of the following. A. Transport substances in the cytoplasm. B. Turgor pressure. C. Photosynthesis. 1.5 Structure H plays an important role in cell Y. Name three functions of this structure. Question 2. 2. Study the following table. 2.1 Complete the above table by writing down only the numbers 2.1 to 2.6 and your answer next to it. Common errors made by learners in examinations. Unable to answer questions based on cell structures and functions. Activities/assessment. Complete the questions given in this lesson. Consolidation. Work through questions on cell structure in past examination papers. Reflect on your learning and understanding of this topic and assess your progress. Values. By studying this section, you will develop your knowledge of key biological concepts.